All right, thank you, everybody. It's a pleasure to be here today and to be able to present our work on Model AD, which is really charged with creating new mouse models for late-onset Alzheimer's disease. This is really part of a big NIH-funded initiative to improve particularly the translatability of mouse models of late-onset Alzheimer's disease. So in the last decade, really, the NIH has established a variety of consortia that interact with each other to both generate and better understand human late-onset Alzheimer's disease, and this is through the AMP-AD consortium, Accelerating Medicines Partnerships for Alzheimer's Disease, and that's well established and has been going for more than 6 or 7 years. But also more recently, the Alzheimer's Disease New Medicine Centers, or TREAT-AD, which are charged with identifying new drugs, both through target validation and drug discovery, in order to then test them in mouse models such as those that are being created by Model AD. Everything I'm going to be talking about today is available either on the modelad.org website, but also all models are made available through JAX Mice, and all data will be deposited in the AMP-AD knowledge portal. And the essence of all this work is really open science. So we get the data out and we get the models available at the earliest opportunity. It's a huge consortium, and I don't have time to name everybody here, but it essentially exists in multiple institutes, including Indiana University, University of California, Irvine, University of Pittsburgh, Sage Bionetworks, and of course the Jackson Laboratory. And I'll be acknowledging 1 or 2 people as we go through this. As I said, it's funded through the National Institute on Aging with our program officers, Susanna Pancheska and Larry Ruffolo. And it's funded through 2 U54s, the first one to the Jackson Lab, Indiana, Pittsburgh, and Sage Bionetworks, and the second one to UC Irvine. What I'm going to be focusing on today is the work being done by our own consortium, so Indiana, Pitt, JAX, and Sage Bionetworks. So the whole essence of the consortium really is to take clinical data and generate more translatably relevant load models. And I'll go into more detail about how we do that, and to translate those phenotypes using both omics, neuropathology, neuroimaging, and ultimately to do drug testing through our preclinical testing core. One thing we were charged with very early on was to standardize the phenotyping of widely used early onset or familial Alzheimer's disease models, and really using these models to establish our phenotyping pipelines. And we chose a number of them, but here I'm highlighting data generated on the 5X FAD model on the B6J background. And so you can see here that we performed in vivo imaging using an amyloid PET tracer. And you can see that amyloid is detected as early as 4 months. Easiest to see here in these cartoons at the end, where you can see the red is amyloid that's depositing. So this is a very aggressive amyloid producer and has been widely used to look at, for instance, amyloid-induced microglial activation. It gets things like astrocyte and microglia activation in both the cortex and the hippocampus, as I said, induced by the amyloid. I'll come back to this in more detail later, but we do a lot of transcriptional profiling because much of the data that's coming out of the AMP-AD consortium is transcriptome data from AD patients and controls. And so we're able to ask not necessarily how similar are our models at the level of what you think of as traditional Alzheimer's pathology, such as amyloid and tau, but more what are the molecular changes that align better with human AMP-AD, and that's going to become key as we move forward. We're also doing blood-based biomarker assessment, and we've done this in the 5X FAD. And again, you can see that you can detect A beta 1 to 42 as early as 4 months. And all of this data has been deposited in the AMP-AD knowledge portal and is available here. And there's also a manuscript in preparation from Adrian O'Black and others. I want to just start at the end in some ways, which is the preclinical testing core, and this is led by Paul Torito at Indiana University and Stacy Rizzo at University of Pitt. The primary aim of Model AD is to generate more clinically relevant preclinical testing models. As you can see, the bulk of the work is being done at Indiana and the University of Pittsburgh, with some contribution from Sage Bionetworks, who are hosting all the data. and a small amount of work still going on at the Jackson lab. More was when Stacy was still with us, but she moved to Pitt. But Kristen Onos and Kelly Geezer and Mike Sassner have been contributing to the work in the PTC. So essentially what the preclinical testing pipeline is a robust and rigorous way of performing preclinical studies, starting with, for instance, a test compound and then doing PK and PD studies, all the way through to then assessing by a particular phenotyping method. In this case that I'm showing here is in vivo imaging, similar to what I showed on the previous slide for 5x FAD. And if you look in more detail at this diagram, what you can see is there's a dose-dependent reduction in amyloid using this drug verabecestat, which is a base 1 inhibitor. So it's as expected, you see this reduction in a dose-dependent manner in males and females in 5x FAD mice. And where the PTC sits is really to take information both from new compounds coming out of treat AD combined with new models through model AD, and to assess the efficacy and the benefits of those compounds in the right model. I want to highlight particularly the Stop AD portal. So this allows community members to nominate the compounds that they think could be tested within the PTC, within model AD. And this is just showing the details. So the Stop AD portal is available, and you can enter a lot of information that you already know about your compound, and then it will get ultimately to an in-person committee review before it's deemed to be a suitable compound to be both for the planning and then the initiation of the testing in the PTC. And so I urge everybody to Google Stop AD, and if they have compounds that they're interested in, to enter those compounds there. So as I said, the ultimate aim of Model AD is to put new preclinical testing models into the PTC, and that's really driven by two additional cores, which is the Disease Modeling Project, or DMP, and the Bioinformatics and Data Management Core, or the BDMC, and this is led by myself, Mike Sassner, Bruce Lamb, and Adrian, and then the BDMC is led by Greg Carter, Lara Mangravit, and Andrew Sakin, Indiana. And again, there's a huge infrastructure behind this, both at Indiana University, the Jackson Laboratory, University of Pittsburgh, and Sage Bionetworks. Everybody working tirelessly to both generate the mice, to phenotype the mice, to identify the variants, and ultimately to get all the data publicly available and the mouse models available through JAX Mice. When we think about creating new translationally relevant load models, we kind of divide it into a number of different aspects. The first one that we chose was to introduce strong risk variants for late onset Alzheimer's disease. So very much getting away from the idea of Alzheimer's disease has amyloid and tau pathology, so let's drive that as fast as we can, for instance. But let's design our models based on the genetics of late onset Alzheimer's disease. Two of the strongest risk factors that we introduced initially was the E4 variant of ApoE, as well as the R47H variant of TREM2. And so we made that, and I'll show you some data on that model. And the second thing was, rather than driving amyloid pathology through mutations in human relevant genes, we just simply humanized the AD genes. And again, I'll come to why we did that, but particularly in APP and MAPT, which are the genes responsible for amyloid and tau. The third one then was to think about late onset Alzheimer's disease as a collection of risk variants that conspire together to increase the disease. And so for this, we need to identify very precise risk variants through leveraging the human AD data that's available, and introduce those into mice on top of what might be a sensitized background, such as a mice containing ApoE and TREM2 and humanized APP. And then to recognize the fact that genetics may not be the be-all and end-all of risk for late onset Alzheimer's disease. There is environmental risk, and we've started some projects that focus on one of the major environmental risks, in this case, diet and obesity. And then finally, at the Jackson Labs, we're all aware of genetic diversity because we focused initially on the C57 Black6J mouse, but for many, many years, we've known that by modifying the genetic context in which these mutations arise, that you can modify the disease outcomes. So genetic diversity is going to become a critical component of Model AD, where we search for the most susceptible strain in order to drive these phenotypes. And then to think about the human relevant outcome measures, such as I've mentioned, transcriptomics, fluid biomarkers, neuropathology, in vivo imaging by PET, MRI, and also behavior and cognition, but really focusing on those that are most translatable right now, particularly transcriptomics, fluid biomarkers, and in vivo imaging. And then, of course, to distribute the resources as quickly and as efficiently as we can through both JAX mice with mouse models available with no imposed restriction, and also all the data made available through the knowledge portal. So here's the first model that I wanted to just go through a little bit of data with, which is the APOE4 TREM2 R47H model. So it's carrying mutations in 2 strong risk genes. We've done a whole variety of assays, including frailty and behavior. And you can see here that as they age, this is a frailty score developed by Stacy Rizzo, which shows that there's no genotype effect here, but as expected, as mice age from 4 through to 24 months, you see an increase in their frailty in both males and females. And also then a cognitive task here the Y-maze percent alternation, and there's no cognitive deficits out to 24 months in these mice either. But what is interesting about these mice is by in vivo imaging, they have cerebrovascular deficits. And this is measured using 2 PET ligands, copper PTSM for blood flow and FDG for glucose uptake. And what this chart is indicating is either a decrease compared to controls in the amount of blood flow or an increase compared to controls in the amount of glucose uptake. And what's really interesting about these mice at 8 and 12 months of age is there appears to be an uncoupling between blood flow, which is on the decline in some brain regions, and glucose uptake, which is on the increase in other brain regions. And so a lot more work now needs to be done to understand how this phenotype may relate to changes in cognitive status if we add on additional variants. In addition, through long-term potentiation tests, this was done at Indiana University, you can see that there's an age-dependent decline from 4, 8 to 12 months in LTP in both males and females. And finally, by protein levels in the brain using mesoscale discovery, you can see an increase in a pro-inflammatory marker, IL-6. By transcriptomics and neuropathology that I'm not going to show you today, you see an increase in inflammation-related pathways, a change in inflammation-related pathways in this model, which we're calling load 1. And also in TREM2 mice, so this may well be driven by the TREM2 R47H variant, again, compared to controls at 24 months. And by neuropath, we don't see evidence of amyloid accumulation. So in terms of a preclinical testing model, the absence of amyloid would suggest that this wouldn't be an appropriate model for testing amyloid-induced effects, but it's beginning to identify some additional endophenotypes, if you like, that are relevant to Alzheimer's disease. And again, all of this work is being put together in a manuscript that you expect to see out in the coming months, led by Kevin Katridis at JAX. So the second set of models I want to describe is the humanizing of APP and the humanizing of tau to better align the mouse to the human. For those of you who know a bit about Alzheimer's disease, you may know that the human A beta sequence is actually different to the mouse A beta sequence in 3 amino acids, as shown here, the Y, the R, the Y, and the H. This is thought to render the human protein more amyloidogenic. In previous mouse models, such as the 5X FAD and the APP-PS1, people have introduced using transgenes, so multiple copies, high expression of mutant forms of A beta. What we've done in Model AD is to move away from that and actually to humanize the mouse A beta sequence within the APP gene to create human forms of A beta. And so this mouse that we're calling load 2 is humanized A beta on load 1, which is the B6J APOE4 TREM2 R47H. So this now carries 3 potential risk variants if you include humanized A beta as a risk. We're working on this mouse. We have early indication of elevation of soluble A beta levels in the brains at 8 to 12 months. but as yet no strong evidence of Phi-OS positive plaques, which would be the kind of the mature forms of the plaque. But we're still aging these mice out, as you can see from the timeline. The third model then would be LOAD3, which is now taking the humanized A-beta ApoE4 TREM2 and now adding in the human MAPT gene. And so again, for those of you that know about Alzheimer's disease, humans, as they post-development, show multiple isoforms of tau, what's known as the 4R and the 3R, and these are in equal ratios, either because of the inclusion or the exclusion of exon 10, and this is shown here in the cerebellum and the forebrain. Now, in mice, you generally only get one set of isoforms, the 4R, and this is thought to lead to the mouse being less likely to develop tau pathology. And so what we've done is humanized, i.e. replaced the mouse tau with the human tau. And this was done by Dr. Michael Koob, and he's provided the mice for us, and we've now introduced it to create this LOAD3 mice, which essentially carries four risk loci, the ApoE4, the TREM2, the humanized A-beta, and now the tau. The timeline for phenotyping, one of the major things of model AD is that we need to get real and we need to do our aging studies. And so all of our deep phenotyping studies are going out to 24 months of age. And so this is a long process. In terms of LOAD2, we've got mice established that are 4 to 12 months of age. And we're waiting on establishing those that are going to go to 18 months and 24 months. And we're just building the cohorts for LOAD3. The 24-month time point was recently added thanks to supplemental funding from the NIH. We call this process deep phenotyping because it's going to be done at both sites, so at Indiana University and JAX, because we want to ensure that if a phenotype is seen on one site, it's seen on another to maximize chances of reproducibility of certain phenotypes. And as I mentioned the phenotyping pipeline that we've developed includes biometrics, blood profiling, neuroimaging, behavior, as well as post-mortem assays such as transcriptomics, proteomics, metabolomics, neuropathology, biochemistry, and fluid biomarkers. And these are particularly designed to align with the data that we have available in human AD, particularly through the AMP-AD projects. In addition, because not everybody is interested in, for instance, TREM2 or ApoE, we're also assessing these mice in singles, so humanized A-beta alone, the tau alone, and also humanized A-beta, tau, and these will be available a long time before we have the phenotyping data. So you'll be able to get these from JAX mice. So the third aspect of this is to take those what we might call platform strains, for instance, LOAD1, 2, and 3, and now introduce those additional risk factors that might be the critical risk factor that tips a mouse over from having some endophenotypes of Alzheimer's disease all the way to having full-blown late-onset Alzheimer's disease with amyloid pathology, with tau pathology. And so we do this via a method of variant prioritization and selection, which I'll show you on the next slide. And then we use CRISPR-Cas9 to introduce those new variants into these sensitized backgrounds. And then we have this process that we call primary screening as opposed to the deep phenotyping, which is using a panel that's been designed in collaboration with NanoString to assess the relevance to human AD. At this point, if the model looks promising, we then go through a go, no-go gate, so a promising model, in other words, a model that looks a little bit more like LOAD with the variant than without, will go through into the deep phenotyping that I described on the previous slide. But if that particular variant didn't seem to do what we hoped it might do, we will then do no additional phenotyping, but the model itself will always be available through JAX. So just to walk you through this process in more detail, then the Bioinformatics and Data Management Core, led by Greg Carter, is taking multiple data sets, and they're listed here, IGAP, ADSP, ADGC, and ADNI, as well as AMP-AD, for instance, to identify variants from all of those studies, and then to prioritize those variants that are most likely to be load risk relevant. And you can see here that you can take the loci that we've so some of the famous ones that we've known, such as BIN1 and CD2AP and SORL1, and identify a potential risk variant. Because if we're going to do CRISPR-Cas9, we don't necessarily want to knock the gene out. We want to precisely alter the gene to be as human relevant as possible. And so you can see here that the criteria for selecting a variant includes predicted function, the pathogenicity, importantly, conservation between humans and mice. So, for instance, one region that we struggle with is the MS4A6A region, which is quite different between humans and mice. And so we're unable right now to identify a risk variant, but are considering humanizing this region in order to include this region because we think it's an important gene. But basically, if we can't find conservation, we won't necessarily introduce that simply by CRISPR-Cas9. And then obviously looking at an allele frequency, because we don't necessarily want to take the rarest alleles, because we want these as to reflect as much as possible the human population. And so by these methods, we can identify specific variants that then get introduced into the mice. And so there's a lot of data that goes behind this slide that I don't have time to talk about. But essentially, what's done in AMP-AD is to identify modules of genes and then annotated those genes that reflect changes between controls and AD patients. And so three sets of modules would be considered relevant to extracellular matrix and immune and neuronal and oligo/astrocytes and metabolism / proteostasis. And then using this nanostring mouse AD panel that has been developed by people like Christoph Preuss and Greg Carter and others at Sage, we can now assay the transcripts of sets of genes that reflect these human modules to ask, in a new mouse model versus a control, does that transcript change in a similar way to how it was changing in human AD? And so what this is basically showing is if a set of genes change in a way that looks like similar to human AD, you'll get a blue spot, and the darker the blue, the higher the correlation. And if it doesn't change, you'll get white, and if it changes in the opposite direction, you'll get red. And so I hope you all understand this, because this is a really cool way of beginning to determine the relevance of different mouse models to human AD. And what you'll see here in 3 variants that were tested on the background of load 1, a variant in ABCA7, in MTHFR, and SNX1, you can see that there's an age-dependent increase in the relevance of these models to human AD. So for instance, in ABCA7, you can see that there's an increase in these blue spots in both oligo/astrocyte relevant modules and metabolism / proteostasis. So this is what we would consider a go by our primary screening. So this model is now entering deep phenotyping as well as the MTHFR model. And what's also interesting is even though some of the spots are in the same place, the annotation of the genes that are driving that correlation can be different. So in ABCA7, you can see protein folding, and in MTHFR, you can see fatty acid metabolism. And so, watch this space for these models. We think they're going to be interesting towards load models. And what we're now making these models on load 2, which is ApoE4 TREM2 humanized A-beta. And all that data will be out through the knowledge portal. So here's the timing. You can see here we've got many variants, up to 24 variants, some of which have been completed and some of which are still in progress. And all of these mice get aged to 12 months to be assessed. We've actually dropped the 8-month time point because it was deemed not necessary. Knowing the changes between 4 and 12 months was sufficient, and we'd rather get through more mice than have 3 time points for primary screening. And the last set of models are currently being established. Officially, Model AD is going to end in year 5, and so the final models will take as old as we can, but there is talk of Model AD being renewed, and we look forward to that. What I would say, and I'll show this at the very end, is all of these models, whether or not you think they may be preclinical models, they're biologically fascinating in terms of the pathways that may be relevant to late onset Alzheimer's disease. So some of these models are already being incorporated into research labs, such as my own. In my own lab, we work on ApoE4, and we work on MTHFR, we work on humanized CR1. So we're better understanding the biology behind how these genes might be impacting the brain in addition to waiting for those models for doing the critical preclinical testing. So I mentioned on that slide that we're also looking at non-genetic factors, what sometimes is called the exposome. And so here we're looking at high-fat diet. And so the pilot that we ran was to put on load one with MTHFR, load one with a different variant, PLCG2, and load two. I went on a high-fat diet, which is the 45% high-fat, high-sugar diet at 1 month of age, and they're assessed at 12 months of age through that same nanostring assay that I described previously, as well as some neuropath and some biochemistry. Along the way, we're assessing metabolic assays to find out whether we've introduced that risk, that environmental risk that we wanted to. And here's just some examples. So for instance, for load two, we see that increase in weight in females. It's the same in males, although a little bit greater in the females, as well as the MTHFR variant on load one. So you can see an increase in this case. I'm showing it in males, although there is a plateau as you get to 12 months. This is actually age on diet. So the mice here are 12 months old. And you can see a plateau where the control diet mice are catching up. And also with this high-fat diet, which has been widely used by many, we see the increase in total cholesterol, which is one of the metabolic features that increases risk for Alzheimer's disease. So the thinking here is that with this preliminary data, we may well then put additional cohorts of mice out to 24 months of age to see how aging by genetics, by environment, in this case diet, modifies AD-relevant phenotypes. And the first data out to 12 months is going to be available fairly soon. I also want to take a moment to talk about genetic context. The vast majority of Alzheimer models are on the B6J background, or at least on a commonly used, inbred strain. And kind of hopefully everybody knows by now that if you move a transgene, if you basically move any gene or knock out any gene on a variety of different genetic backgrounds, you're liable to get a different result. And so the consistency of phenotyping across the mouse strains, it's really important to understand. In my lab, work done by Kristen Onus and particularly Stanley Yang as well now, have shown that if we move the commonly used Burchell transgenes, which are mutations in APP and presenilin, which ordinarily cause a lot of amyloid deposition by 6 months, not a whole lot of cognitive deficits, using robust cognitive assays out to about 12 months, and little neuronal cell loss on the B6 background. If you move those to, say, for instance, a wild-derived mouse strain, this is our favorite wild-derived strain at this point, WSB, you see changes. In this case, I'm showing IBA1 in white, and which is lighting up microglia, and GFAP in green, which is lighting up astrocytes. You see changes in the amyloid response from B6 to WSB, and if you go and read the paper, you'll see that this actually has much more cerebral amyloid angiopathy than on the B6 background, and CAA is a common feature of late-onset Alzheimer's disease, particularly in the later stages, and we see more neuronal cell loss in WSB APPs than we do on B6. So we kind of know that by moving to what may be a more susceptible strain, WSB, that we may well be driving the load phenotypes. So in Model AD, what we've done is leverage the collaborative cross strains, which is essentially an 8-way cross of B6 WSB plus these other 6 strains, and they've been randomly mixed. So in this column, you can see CC line number 6 versus 41, and the allele for a particular gene is colored based on the strain that it inherited. So here you can see ApoE from NOD, BIN1 from a variety of different strains. And what Greg Carter has done is to identify CC lines that vary greatly in genes known to be impacted in late onset Alzheimer's disease, such as ABCA7, ApoE, TREM2, SOR1, etc. And if we look at their gene expression data in the hippocampus, work done by Alyssa Chesler, and we can see that their gene expression changes. So for instance, CD33 has high levels in CC6 and 41, but lower levels in CC2. So the hypothesis here is that by varying the transcript levels of load relevant genes, we may well modulate the phenotypes that we see when we then introduce stronger risk variants. So the experimental design for this study is to vary the genetic context in the presence of amyloid. To do that, we're taking the CC lines and making F1s with ApoE4, mice that are homozygous for ApoE4 and hemizygous for APPPS1. So every mouse will be F1 for B6 and the CC line, have one copy of ApoE4, and either inherit or not the APPPS1 transgene. So this gives us litamate control at the same time. We're analyzing this by RNA-seq, and that's right now being done in Greg's lab, and then we'll add NeuroPath to look at things like what does amyloid look like? Have we changed amyloid and glial responses in a way that we've seen previously? And again, the idea here is by saying, can we generate a mouse strain that shows stronger AD phenotypes, and then translate this work from the early onset transgenes such as APP to the late onset risk variants that I've been describing. So just in the last couple of slides, I just wanted to highlight how we're also thinking more about how we're going to get these mice into preclinical studies, and one of those is to focus on biomarkers. And so we're analyzing both cerebrospinal fluid as well as blood-based biomarkers. And so we're looking at proteins using mesoscale discovery as well as other methods such as quanterics. And in this case, for instance, LOD2, which is ApoE4 TREM2 humanized A-beta, we're beginning to see an increase in neurofilament light in the cerebrospinal fluid at 12 months, which is kind of exciting and suggesting that LOD2 may well have some neuronal cell changes that may well get exacerbated as we take them out to 24 months. And we can detect that obviously in CSF. And in terms of blood-based biomarkers, I've mentioned this previously, but also we're doing the amyloid, and we can see this. For instance, we can detect amyloid in the blood in the 5X Fab. It's kind of an aggressive early onset model. You can see it as early as 4 months here. So additional biomarkers that we're working on, we're looking at phospho-tau. We're looking at synaptic markers, because if anybody attended AAIC, there was a lot of interest in seeing synaptic markers in particular the CSF. And so we can track synaptic markers, for instance, PSD95, which is a post-synaptic marker, as well as astrocyte markers such as GFAP. The strains that are being tested, LOD2, LOD3, as well as those that I talked about that passed primary screening, such as LOD2 ABCA7 and LOD2 MTHFR, as well as a strain of high research interest, which is LOD2 carrying the PLCG2 risk variant M28L. And then finally, I just want to show that we're constantly evolving our pipelines, our phenotyping pipelines with these additional assays. So Mike Sassner is working with tissue vision to be able to do amyloid imaging, so to identify regional deposition of amyloid, and in this case, it's in the 5X Fab. So you can basically get 3D reconstructions having injected these mice with methoxy 04, and it sticks to the plaques, and then you image them, and you can identify where the amyloid is. And this is important because obviously amyloid doesn't necessarily deposit throughout the brain all at the same time. There are regional differences in humans, and we want to know how similar our mouse models are going to be. Similarly, spatial profiling with nanostrings. So this is digital spatial profiling. So we can look at different regions such as the subiculum and the cortex and identify across genotypes whether some genes are changing, some proteins are changing, whereas not changing in other regions. And you can see here a panel. We're working with Eric Bloss, who's a new investigator at JAX, who can do high resolution synaptic imaging. A lot of people think the critical component of Alzheimer's disease is the loss of synapses. And so we need to improve our synaptic imaging, particularly if we're going to be assessing biomarkers in the CSF that will be related to synaptic proteins. So we're piloting with Eric array tomography, which can get high resolution imaging looking at synaptic organization. And then finally, sleep or EEG. This is being done by Stacy Rizzo at Pitt. And you can see here one example where there's sleep changes in the inactive state in LOD2 mice. So another hint that we think LOD2 at 12 months may well be beginning to get phenotypes relevant to late onset Alzheimer's disease. But we need to do more work on understanding LOD2, and that's ongoing. And just some highlights of you know, how you assess the success or otherwise of Model AD through the PTC. They've validated two compounds, and they've done a whole load of in vivo imaging, more than 100,000 imaging data points. Interactions with Treat AD, which will be critical because Model AD as a consortium is interacting a lot with AMP-AD, and as I mentioned, Treat AD. In terms of mouse models, I've talked through 4 base models that we've created. We've created models for 31 different mouse models in addition through the conserved coding variants as well as non-coding variants. And we've got a lot of phenotyping in progress and a lot of data that's already been deposited in the AMP-AD knowledge portal. And for those who want to know everything about all the models we've created, here's the list. The base models, we've got allelic series of ApoE, allelic series of TREM2. We've got APP and humanized APP and tau. And we've got these platform models that we're using, the load models 1, 2, and 3, the conserved coding variants. And if you see your gene of interest or your pathway of interest, I urge you to work on some of these variants, as well as knockouts. When you do a CRISPR-Cas9 study, you often get the knockout as well, and so we keep those knockouts. We've humanized some loci, including humanized CR1, and we're already beginning to think about non-coding variants because not all of the risk loci will be nice coding variants to make. We have to make non-coding variants. And then finally, publications. You can see that we're not only making our data available early, we're also publishing it. So we've got 9 manuscripts already accepted. We've got a whole variety of manuscripts that are in preparation. Much as this, we give oral presentations to promote our work because we need these models to be used in research labs. And it's cool to think that 60 labs have already ordered mice as of September 2020. So we're super excited. And then finally, I wanted to highlight the work going on in my lab. Anybody here in red is benefiting from models that were made in Model AD. And so I mentioned MTHFR and ApoE4. So particularly Elena Reagan, a postdoc in my lab, is working on the mechanism of MTHFR. It increases risk for cerebrovascular damage. And Kate is working on ApoE4, looking at the interactions of genetic risk with aging, with exercise. And then the collaborators, Mike Sassner, Greg Carter, who are partners in crime in Model AD at JAX, as well as other PIs that are benefiting from mouse models we've made in Model AD, such as Jennifer Trowbridge and Eric Bloss, who were working on humanized CR1. External collaborators, funding, the NIA, National Eye Institute, the Alzheimer Association, and more recently, the Bright Focus. And I have a postdoc slots available, so if anybody's interested, please feel free to get in touch. And with that, I'll end. Thank you very much.